Hello, this is Mr. Russell, your AP Biology teacher. Today we're going to be continuing our discussion on biochemistry, and in particular we're going to be continuing our discussion on macromolecules. Remember earlier we talked about how macro means large, so we're talking about large molecules. Today in particular we're going to be talking about proteins. These are going to be also known as polypeptides. And we're going to be talking about enzymes. Enzymes are a type of protein. Well, these macromolecules make up greater than 50% of an organism's dry weight. If you remember from earlier in our discussions, we've talked about the dry weight of an organism is referred to its biomass. Well, we also talked earlier about the ending of names, such as ose. If something ends in ose, it is likely a sugar. Sucrose, maltose, lactose, fructose. Well, if we continue that, we look at our suffixes. If something ends in lin, L-I-N, like insulin, then it's going to be a protein. If it ends in ase, then it's going to be an enzyme, such as sucrase. So if we think about sucrose, a sugar ending in ose, and sucrase is an enzyme, well, sucrase is likely to be the enzyme that promotes the dehydration reaction for sucrose. Now, if we think about, we talked a lot about monomers and polymers, and we know that monomers are building blocks. And in this case today, we're going to look at the building blocks that we're going to be calling amino acids. So there are 20 different types of amino acids that can be involved in making proteins. Proteins and enzymes usually have hundreds to thousands of amino acids in their structure. Well, amino acids have 4 different parts to them, and that's what I want us to look at here today. So if we look at this first part, this is going to be that carboxyl group. Here we can see that double bonded carbon and oxygen that's going on there. This is going to be the part that acts as the acid just because it can give off this hydrogen down here. Now, also we've got an amine group, or NH2. This NH2 group is going to act as a base because it can accept the third hydrogen. Then we got right here in the center is going to be this alpha carbon. This is the central carbon that's going to hold the whole molecule together. Right up here at the top, and sometimes it can be flipped over, maybe on the bottom. So in our next slide, you're going to see where it is going to be on the bottom. This is going to be referred to as the R group. This R group is the most important part of this whole structure because it gives each amino acid its distinctly different property. And we're looking at this, we know that amino acids are going to have distinctly different R groups. So if we look at our structures here, and we look where we said that our R group is flipped, and we're looking at here in the white. There is our R group. It has an H. Everything else about the structure is the same in all of these molecules except for the R group. If you notice, it changes a little bit in each of these. And as it continues to change, it builds. It gets bigger. If you notice, the names change. You went from glycine to alanine to valine to leucine. And it continues to build because it is the R group that makes each amino acid distinctly different from one another. Individual amino acids, or monomers, are bonded together by a covalent bond called a peptide bond. An amine end of one amino acid is positioned to combine with the hydroxyl end of another. So if we look here, you've got your amine end, the NH3, or the NH2. In this case, it's the NH3. So it also is lined up right there at the end of our carboxyl end of the second amino acid. So what happens is these will join together here and here. So, and then as they continue to join, then you end up with larger molecules forming those polymers that we were talking about earlier. Now, how does this happen? If you remember, we talked about the open bonds that get left behind. this happens by removing that water in those dehydration reactions. So it allows for a bond between the carbon and the nitrogen to be created. When we put many amino acids together, we end up forming the polypeptide or the protein that we see here. So if you look where it's got the 1, 2, 3, and 4, here we're looking at a polypeptide has been put together because of the dehydration reaction that has been formed. Now if we want to break that bond, all we got to do is put our water back in. It breaks the bonds apart to go back to individual monomers. Now, if we look at peptide bonding, here again you can see those individuals being broken apart so that we've got our dehydration reaction. This is probably a better image of our dehydration reaction. As you see there, there's your peptide bond there that forms as the water gets removed from the system. Now, we think about the arrangement and the quality of amino acid, its affect the structure and function of that protein or enzyme. So if you think about structure is going to equal function. You're going to see structure and function over and over and over again in AP Biology. Probably on the AP exam they're going to have you to discuss the structure and function of something. They're going to give you a structure. They're going to say, talk about its relationship. It could be organelles. It could be anything. Who knows? We'll get into that more as we go. Now let's look at this first one here. Think about a primary structure. You've got to know these because these primary, secondary, tertiary, and quaternary structures are going to get repeated throughout this whole year. Well, this is represented by the symbol right here up at the top, this 1. This is going to be your primary sequence or primary structure. Now, so the primary structure just refers to the sequence of bonded amino acids. So think sequence for primary structure. Here you can see lots of these little amino acids. You get one here, one here. How are all these linked together individually? That's going to be your sequence. What is this sequence in order? So if we took this and we rearranged them, well then we're changing the sequence of it. So our primary structure has changed. Now, why is that important? Well, if we look at the structure and function of our blood and how it carries oxygen, primary sequence is really, really important. So just look at the difference between sickle cell disease and normal red blood cells. Just by changing this sixth, let me go back here. Just by changing this sixth, I did it again, sorry guys. Just by changing this sixth amino acid here, changes our whole structure from something that can carry normal red blood cells to being this horrible, horrible disease. The easiest way to remember when you're thinking about sickle cell is knowing that it's the sixth amino acid that gets changed. If you think about the mark of the beast or the mark of the devil being 666, that's a bad number, right? Well, the bad number in your primary structure, primary sequence, ends up equaling a really, really bad disease. Now, let's look at our secondary structure. Here again, secondary structure often symbolized here. So when we're looking at these secondary structures, we're talking about how hydrogen bonds allow for overlapping and coiling to occur in the folding of the protein into this into a 3D shape. All proteins must be folded in order to work. You need to think about folds and coils when you're talking about secondary structure. And when you think about coiling, it's referred to down here as the alpha helix. So here we are down here. This is our coiling. We think about overlapping. This is going to be these beta pleats here. So go back and take a look at this. Research a little more. Look at other videos. Look at your book, look at any of our notes, and look at coiling, alpha, helix, and overlapping, where this folding is going to be our beta pleats here. Now, look at our tertiary structures. The T-E-R-T, the tert, means third. So here we're looking at these disulfide bonds. Di meaning two, sulfide means it's got sulfur in it. So we've got disulfide bonds, and what it's going to do is it's going to help to build this bridge right here to be able to allow these proteins to fold. In order to make this disulfide bridge, the amino acid, do you see an amino acid on this anywhere? Let me clear this off for you. Do you see an amino acid? You should see one right there. Well, there's your amine group that kind of gives it away. So when you look at this big part here, here's your amino acid, which is going to be cysteine, is going to be needed to help to form this disulfide bridge in your tertiary bonds. Now, also you have what's called a quaternary structure. This is when two or more polypeptides are woven together. Think about hemoglobin or red blood cells. They have 4 proteins woven together to be able to make it. So when you're thinking about quaternary, you need to think about multiple woven together for quaternary structures. In this case, you can see you get these structures. Here's an individual structure. Well, when these get combined together, then in this case, this one's going to be carrying some blood. This one's going to be carrying blood. This one's going to be carrying blood. This one is, and this one is too. And these are going to be healthier cells. The problem comes in when these structures don't carry blood like they should. And we'll look at that here in just a second. Here we're looking at protein in its natural form, which is just going to be carrying out its functions. Depending on how it folds and coils, is going to carry out the different function of it. Now, if we look at this and we look at our primary structure up here, remember we said how it was this sixth, oops, let's see, sorry guys, okay, let's try this again. Okay, remember we said it was this sixth sequence right here that made the big difference? Well, if you think about structure and function, here we're looking at the primary structure being up here. What is it made of? What is its structure? What causes everything to be made? What's these amino acids and how they line up together that forms the structure? If we look at the secondary and tertiary structures, well, here we get this beta subunit in the secondary and tertiary structures. Well, over here when we're looking at sickle cell, it changes a little bit. We get this exposed hydrophobic region that doesn't like water in the beta subunit. Well, let's come down here in this next little part, our quaternary. Here we're looking at normal hemoglobin. This is a top view of it. And over here we're looking, over here to the right, you're looking at sickle cell. You can see the differences. They don't even look the same at this point. Well, the problem is when we start looking at the function of the primary, secondary, tertiary, and quaternary, their function when they all work together, if you can see down here at the bottom, totally looks different. So therefore, they're probably going to carry out different functions. So your molecules are not associated with one another. So therefore, each individual molecule is going to carry its own oxygen. Well, if we look over here at our sickle cell, the molecules are interacting with one another, and they're going to crystallize into a fiber. So therefore, the capacity to carry oxygen is greatly reduced. Huge, huge problems for you. Difficulty exercising, creates a lot of pain, long-term health effects, very, very painful disease. Okay, let's look at denaturing. Denaturation. We're talking about how enzymes unfold. This is the protein unfolding. It unravels. So you think about how if something is becoming denatured, it's coming apart. So this unraveling of a protein or enzyme causes it not to function as it should. So the denaturing can be caused by change in pH or acidity. It can be changed. It can be caused by changes in salt concentrations. It can be caused by changes in temperature. These are things that you're going to see over and over in this class. So you've got to know that what causes a protein, what has a huge impact on the function of a protein, is going to be things that might cause it to denature. The pH change, the salt concentration change, and change in temperature. These are all going to speed up and slow down reactions. Now, we have this term called a chaperonin. This is a protective structure that allows proteins to fold inside of the structure without water being present. Now, think about a chaperone. What does a chaperone do? It's a person who watches over a function or a date to be able to make sure nothing terrible happens. It's kind of like being at one of the high school dances after a football game. There's chaperones there to make sure everything goes like it's supposed to. So that's the purpose of this in protein folding. Now, we think about nitrogen cycling. The majority of nitrogen is removed from the air by water. Remember, water is a universal solvent, so the gas is dissolved in the rain or snow. The nitrogen in the water mainly is consumed by nitrogen-fixing bacteria. It's found in the soil. It converts into ammonium, NH4. This process is referred to as nitrogen fixation. So the ammonium can then be absorbed by plants to help make proteins in DNA or RNA. So some ammonium, the NH4, in the soil is also consumed by nitrifying bacteria and converted into nitrite, or NO2, first, and then ultimately into nitrate, NO3. The process is called nitrification. So the nitrates are also absorbed by the plants just as the ammonium was. So the plants ate the nitrates.